Hi guys, welcome back. It's week 3. We are on neoplasia now, or neoplasia, I should say. This PowerPoint is really an overview and a review to complement both the PowerPoint that's already uploaded into the module, as well as to complement your readings, because this really follows the outline of your chapter. I know this book and its content can be a little overwhelming, and I don't want to bombard you with too many PowerPoints. This PowerPoint really just has some summarizes a lot of the most important points that your text points out, as well as things that I thought were important for you to understand from a clinical standpoint. So, cancer is obviously a big thing that we need to understand as clinicians in order to be able to not only treat and diagnose, but educate our patients on. So things that you should be aware of are really made more clear in this particular PowerPoint. I did it both visually from images as well as some really good videos that I thought were very helpful in supporting your text. Keep in mind, in the United States, there are about 163 deaths per 100,000 persons each year due to cancer. Cancer is the second leading cause of death in the United States behind heart disease, and as much as 42% of the cases of cancer in the United States may be preventable with lifestyle changes. Some cancer-causing infections can be treated with vaccines or antibiotics. Screenings can catch cancer early or identify abnormal cells. And throughout this presentation, I'm going to go through an overview of cancer pathophysiology, some important points about the cell cycle with cancer involved. We're going to talk about properties of neoplasms and how we stage and diagnose them as clinicians. Then we're going to follow about the importance of genotyping in use of both cancer diagnosis and treatment. And then we'll just talk about some considerations in the lifespan of cancer, including pediatric considerations and gerontological ones. So here we go, guys. So first things first, let's just do an overview of cancer pathophysiology. What exactly are we looking at? Well, cancer hallmarks are things that will really differentiate cancer cells from normal cells. So the first thing you want to look at is intracellular. You want to look at self-sufficiency. Does the cell promote self-replication? Number 2, ignore growth suppressors. There's no time for DNA repair in cancer patho. Number 3, it evades apoptosis, which means it survives DNA damage. Okay, so that's a problem. Number 4, immortality. It perpetuates DNA damage. Number 5, it has altered metabolism. Number 6, extracellular, on an extracellular level. Number 6, angiogenesis. It supports nutritional needs. Number 7, there's invasion and metastasis. It crosses anatomical boundaries. Okay. And number 8, evasion of the immune system. It ensures self-survival. All right, and this is a great diagram. So you have 1 and 2 right here. They're moving through the cell cycle without stopping for DNA repair. Number 3, we're evading apoptosis. Okay. Number 4, where's number 4? Oh, here it is. We're going through the immortality phase. It's perpetuating DNA damage. Number 5, we're altering metabolism in the mitochondria, right? So we're going through aerobic glycolysis. Number 6, we're going through angiogenesis. Okay. Number 7, invasion and metastasis. And then, of course, number 8 right here, we're evading the immune system. So that is how cancer, that is the pathophysiology of cancer in a nutshell. So here's what we're looking at in terms of a normal cell versus a cancer cell. This is the difference. In the normal cell, we have a large cytoplasm, a single nucleus, a single nucleolus, and fine chromatin. And over here, it's the opposite. We have a small cytoplasm, multiple nuclei, multiple and large nucleoli, and a coarse chromatin. Now, The cell cycle in cancer, again, this gets very technical, so I always find videos with animation really helpful, especially when it comes to the cell, to just go over the main points that we need to really focus on in order to understand how the cell is changing within cancer. So you're looking at interphase, we're looking at the mitotic phase, very important. We're looking at the control of the cell cycle and the checkpoints, and it'll go over all of that in this video.
Cell cycle in cancer. Cancer can develop in any of the body's tissues and at any age. The clinical presentation is variable. However, cancers share a common developmental principle, a gradual acquisition of errors in genes that are important for cell division. In this episode, we will cover the basics of the cell cycle, important features of cancer cells, and their stepwise transformation. Let's start with a general reflection of the human body and its cells. The cells of the human body are very small and not visible to the naked eye. Therefore, it is difficult to comprehend the total number of cells in the human body. The current accurate estimate is 30 trillion cells in the body. If the cells were lined up, they would stretch over approximately 1 million kilometers. In other words, the cells would circle the Earth approximately 30 times. However, this amount represents the number of cells present at any given time. As cells age, they are continually replaced. Within the average human lifespan, the body produces approximately 1,000 times more cells than the number of cells at any given time. All of these cells are derived from pre-existing cells by division, which is tightly regulated by the cell cycle and its control mechanisms. Loss of cell cycle control leads to uninhibited cell division and cancer development. Let's start by having a closer look at the cell cycle and its phases. Cell division results in the formation of two daughter cells from a single parent cell over several phases. The cell cycle is composed of 4 phases: the G1 phase, the S phase, the G2 phase, and the M phase. The two most important phases are the S phase and the M phase. During the S phase, or synthesis, DNA is replicated. During the M phase, or mitosis, the cell divides into 2 daughter cells. The G in the G1 and G2 phases stands for gap because these phases correspond to intervals between the M and the S phases. Let's go through the normal sequence of events in the cell cycle. In the first phase, the G1 phase, the cell grows and synthesizes the proteins required for DNA replication. In the S phase, the amount of DNA in the cells is duplicated. In the G2 phase, the cell prepares for mitosis. During this phase, additional proteins are synthesized. The final phase is the M phase, in which cell division occurs. The chromatids are separated evenly between 2 daughter cells. Depending on the nature of the cell, there are 3 options after mitosis. If further cell division is required, the cells re-enter the G1 phase and prepare for the S phase. If further cell division is not required, but potentially at a later stage, the cell may enter a resting phase, the G0 phase. Once in the G0 phase, the cells can exit and re-enter the G1 phase. This is regulated by external signals such as growth factors, which bind to receptors and initiate an array of signals that trigger cell division. However, in most cases, the daughter cells make an irreversible exit from the cell cycle and differentiate. The process of cell division is prone to errors, and it is important that no genetic errors are passed on to the daughter cells. To deal with such errors, the cell cycle has checkpoints at certain stages. These checkpoints ensure that the requirements for the next phase are fulfilled. At the end of the G1 phase, the cell is examined for any DNA damage and whether necessary substrates are available for DNA replication. If necessary, the cell cycle is stopped to repair any damage. In the G2 checkpoint, the cell is examined for incomplete DNA replication. If this is the case, the cell can initiate programmed cell death, also termed apoptosis, to prevent damaged genes from being passed on. The M checkpoint, which occurs during mitosis, determines whether the chromosomes are about to be correctly distributed. All of these checkpoints are necessary because errors are frequent and the checkpoints ensure the integrity of the cell's genome. The cell cycle is internally regulated by proteins called cyclins. They determine whether a cell transitions into the next phase. Cyclin activity is influenced by proteins such as p53 and retinoblastoma protein, which can halt cell cycle progression. In cancer cells, the function of cell cycle checkpoints is usually limited. This leads to uncontrolled cycling, even in the presence of DNA damage. Cancer cells are also independent of growth signals and initiate the cell cycle, even though cell division is not required. In the next slide, we will take a closer look at the development of cancer cells. The uncontrolled division of cancer cells may develop after several characteristics are acquired, allowing the cell to become neoplastic. 10 hallmarks of cancer have been postulated to describe these changes and are shared by most cancers. We would like to highlight 5 of these hallmarks in this episode. First, cancer cells are capable of sustaining proliferative signaling in the absence of growth factors.
5 o'clock.
In other words, they are able to enter the cell cycle without requiring positive external signals. Second, cancer cells are able to evade growth suppressors. Let's use an example here. Non-tumorous cells grown in a petri dish would proliferate until a single layer is formed that covers the bottom of the dish. A phenomenon known as contact inhibition would then prevent the cells from further proliferation. In contrast, contact inhibition is lost in cancer cells and results in uncontrolled cell growth and proliferation, producing a multilayer of cells. Another example of growth suppressors are the cell cycle checkpoints, whose function is usually impaired in cancer cells and leads to unchecked growth. Third, cancer cells can divide indefinitely. They have limitless replicative potential. This is in contrast to non-cancerous cells of the body, which are incapable of indefinite division. The mechanism behind this lies in the telomeres, which shorten with each cell division. However, in germline cells, where repeated division is required, the enzyme telomerase is able to elongate these ends, thereby enabling indefinite cell division. The interesting part? Telomerase is often reactivated in cancer cells, allowing them to divide indefinitely. Fourth, cancer cells also show increased genetic instability and therefore have a higher frequency of mutations. This occurs as a result of the inefficient repair of DNA damage during the cell cycle and does not induce programmed cell death. Fifth, another hallmark is the disabling of apoptosis. As a consequence, damaged cells are not neutralized. In other words, cancer cells evade programmed cell death. The acquisition of these hallmarks and other cell characteristics are not the result of a single event, but the accumulation of DNA damage over time. Let's take a closer look at the relevant genes. It is important to note that mutations can occur in all genes. However, in several genes, mutations will lead to tumor formation. These genes can be differentiated into 2 groups, proto-oncogenes and tumor suppressor genes. Proto-oncogenes are genes whose uncontrolled activation contributes to tumor formation. If the gene is mutated, it is called an oncogene, and cell division is facilitated. Oncogenes are, for example, involved in transmitting growth signals, such as the receptor for the human epidermal growth factor, HER2/neu. In contrast, tumor suppressor genes require deactivation to contribute to tumor formation. The most important tumor suppressor genes control the cell cycle and, when functional, inhibit uncontrolled cell division. One example is p53. It has been nicknamed the guardian of the genome due to its importance. Regarding the terminology, you might want to keep in mind that anti-oncogene is sometimes used synonymously with tumor suppressor gene. Mutations in these genes usually occur over time and will be discussed in the next slide. Tumor development is the result of DNA damage in the cell. The cell's own repair mechanisms usually remove any defects. If the defect cannot be repaired, it is inherited by the daughter cells. In the latency period, further mutations accumulate in the oncogenes and tumor suppressor genes in the cell line. Because cancer formation has not yet occurred, this period is termed latency. Latency can lead to progression. That is, the cell line undergoes malignant transformation and its division is markedly amplified compared to the cells of origin. To understand the dynamics of tumor formation, it is important to realize that mutations are not inherited in a predetermined order from the original cell to the tumor. There is no sense of direction. Instead, cancer formation is the result of random mutations that can affect all genes. Some of the mutations affect cell division and control mechanisms. Such cells divide more frequently and have a growth advantage compared to other cells. In these cells, the probability of a new mutation is elevated because of its increased rate of cell division and absent control mechanisms. On the one hand, mutations in cancer genes are a random event. However, the risk of further mutations increases over time, and malignancy is more likely after acquisition of certain hallmarks. The image shown here illustrates a further feature of tumors. Tumors are not composed of a large number of one exact type of cancer cell, but are a diverse population of cancer cells. This is sometimes referred to as tumor heterogeneity.
So that was a great review. So they talked about the cell cycle, and then they talked about the final phase, the M phase, the mitotic phase, and that's a really important phase of the cell, phase of the cell cycle. So I wanted to just reiterate that in this short video. It's only 7 minutes,
Hi guys,
but it's a
Mr.
great visual
Polybiology here
view
with
of it.
another much requested video, this time on mitosis. This includes the cell cycle and cancer. This is for AS level Unit 2 students studying the AQA biology spec. Here's our objectives for this little video. We're going to understand why mitosis is necessary, describe the stages of mitosis, and recognize cells in different stages of this process. So in my meiosis video, I compared meiosis with mitosis and whatnot. And we said that both of these are forms of cell division. And I like to compare them in terms of copying stages, division stages, how many nuclei we get at the end, and the genetics of those nuclei. So same slide again, sorry guys. Mitosis, one copying stage, one division stage, two nuclei produced, and they're diploid, which means they've got the full complement, the full chromosome number. And they're genetically identical to the cells that they came from. In meiosis, however, there is only one copying stage, just the same, but this time we've got two division stages, four nuclei produced, and they are haploid, the gametes that are produced. So that's the little cheeky comparison, but now we should move on and just focus on mitosis. So why is it important? Well, it produces identical cells for growth. It's also used to quickly differentiate, or rather quickly replicate, cells that have differentiated. So you've got a really quick way of producing lots of identical cells for tissues, for example, like muscle tissue or blood tissue or nerve tissue or any tissue. It's also really useful for repairing damaged cells. But now we know why mitosis is important, we can go on and look at the 5 main stages of mitosis. So they are interphase, prophase, metaphase, anaphase, and telophase. Now, interphase is where we usually start. This is by far the longest stage of mitosis. Most cells will be in this phase for the majority of their life cycle in the cell cycle, and then the other bits are really tiny amounts of the division part. So interphase first, prophase, then metaphase, then anaphase, then telophase. And you guys need to know exactly what goes on at each of these stages. So we'll start at the beginning, and to do that we need a cell. So here's my cell diagram. We should probably just explain, this is very abstract, what are the different bits. There's a cell membrane, a nucleus, the nuclear membrane, the nucleolus, and the cytoplasm. So let's start at the beginning. Let's start with interphase. So in interphase, there are a bunch of little substages. So you've got the first growth phase, and this is where the cell basically produces all of its proteins that are going to be used to build the organelles. So that's the G1 phase, where the proteins are first made. The second stage is the synthesis phase, or the S phase. This is where the DNA is replicated. And finally, in interphase, we have the second growth phase. And this is where the organelles grow and divide, and they increase the energy stores that are going to be used up in cell division, in the other part of mitosis. So that's interphase, and like I said, this is by far the longest stage. Next up, we've got prophase, and we've got a bunch of things happening here. You'll notice the nucleolus has disappeared. Also, the nuclear membrane is disintegrating, and we start to see our genetic information, chromosomes, becoming visible. So here we are, here's 2 chromosomes, each made up of 2 pairs of sister chromatids. Really important that you can differentiate between chromosomes and chromatids. It's something that quite a few students fall down on. So that's prophase. Main thing you're going to see here is the nuclear membrane going away and the chromosomes becoming visible. The next stage is metaphase, where the chromosomes line up along the center of the cell. Apologies for the sketchy animation there. So chromosomes are going to line up on the equator in metaphase, and we get formation of these things called spindle fibers. And the real purpose of these is going to become apparent in the next stage, which is anaphase. So in anaphase, those spindle fibers, they contract and they pull the chromatids, the sister chromatids, to opposite ends of the cell. So what was once a chromosome, two halves of that chromosome, the chromatids, get pulled to opposite ends of the cell. Finally, we've got telophase. Again, sketchy animation, sorry. So we can no longer see the chromatids. The nuclear envelope reforms, the nucleolus reforms, and the spindle fibers disintegrate, and we get the cell division itself occurring. This is called cytokinesis. And if we look at the whole lot of cell division, the whole cell cycle, we see that basically interphase is massive. So we've got the G1, the first growth phase, the synthesis phase where the DNA is copied, the cells preparing for division, which is the second growth phase, and then only about 25% of the cell cycle is actually the mitosis, the division phase. So one thing that the examiner is quite keen on is your ability to recognize the cells in different stages. They might show you an electron micrograph, or even maybe not an electron micrograph. Yeah, I suppose we can't really see a great deal of detail under a light micrograph unless it's quite high magnification. But here we go. There's some diagrams and there's the actual pictures that they came from, the micrographs that they came from. Can you recognize the stages? Well, in this case, it's quite nice because they're in order from this nicely porched Google Images picture. So we've got interphase, where we can't really see anything. We have prophase, we have metaphase, we have anaphase, and finally, telophase. So one other thing that we should probably talk about is the big C, cancer. And what causes it? Well, cancer is caused by damage to genes which regulate the cell cycle. And this means that cells are going to divide and divide and divide uncontrollably, and the cells start to become quite irregular. And these abnormal cell masses, they're called tumors, and they're going to deprive your normal cells, your regular cells, of oxygen, of glucose, of all the things they need, because they're quite selfish like that. And that is essentially cancer. So, in summary, mitosis is really important because it allows the growth and replacement of cells. It produces genetically identical daughter cells. The 5 stages and what happens in them you need to know. They are interphase, prophase, metaphase, anaphase, telophase. And finally, cancer is uncontrolled cell growth, leading to tumors. I hope that's been useful for you guys. Thank you very much for watching. Please like, comment, and subscribe.
All right, awesome review of M phase or mitotic phase. And after you watch it, then you realize why it's so important to understand that phase of the cell cycle in order to understand what's happening in the cancer. Okay, next, your book goes into properties of neoplasms. Definitely know your tumor terminology in terms of the different hallmarks and different types of genes that are within the characteristics of a cancer cell. This is just a great little overview of what each phase has and what's going on in each one. So you should definitely know this in terms of angiogenesis, resisting cell death, replicative immortality, proliferative signaling, evading growth suppression, and metastasis. Okay, clinical aspects of neoplasia. So your book gets into cancer manifestations, the treatment sequel, biological aspects of gene mutations and cancer risk factors, viral causes of cancer. Another very important clinical aspect of cancer that you need to know about as a practitioner is the staging and grading of cancer. In this short video, they do a beautiful, clear job of differentiating between staging and grading. the reasons for staging cancer, and the systems for staging. Now, obviously, if you have a patient with diagnosed cancer, they're going to be referred to an oncologist. However, as a practitioner and as possibly the primary care practitioner of your patient, they're going to always be coming back to you. So you want to know how to read and understand any follow-ups that come from the oncology department or from the oncologist. So you want to make sure you understand what you're reading when it comes to staging and grading.
Stage versus grade. In this video podcast, we will discuss important principles regarding the staging and grading of cancers. By the end of the presentation, you should understand the difference between stage and grade, the reasons for staging cancers, and the systems for staging cancers. Stage and grade are different ways of classifying cancers. They are not the same thing. Grade is a pathological diagnosis, a reflection of how abnormal the cells look under the microscope. In cancer, cells become deregulated and proliferate abnormally. This is called dysplasia. As dysplasia develops, the cancer cells lose features of their tissue of origin, become less and less differentiated, and are presumed to be of higher grade. When we stage cancers, we give an indication of the degree of spread. Understanding the degree of spread has advantages for us as oncologists and physicians because it allows us to guide the questions we ask, examine patients appropriately, order the right tests, and determine in part what their treatment will be. We stage cancers for several reasons. Number one, it gives healthcare providers a common language to describe the extent or severity of cancer. Number two, staging cancers helps to guide treatment. Number three, it allows us to estimate prognosis. Number four, it allows us to compare our results over time. And number five, it allows us to have clinical trial standardization. The types of tests used to determine stage depend on the type of cancer. Tests can include physical exams, imaging studies like CT scans or MRI scans, lab tests, and pathological or surgical reports. Stage is commonly done by the TNM staging system. This is not the only staging system, however, but is the most common. The T is the tumor stage, the N is the nodal status, and the M represents the metastases. With respect to the T stage, as I said, T refers to the extent of the local and primary tumor growth. T0 means there's no primary tumor that can be assessed. TIS stands for carcinoma in situ. And T1, 2, 3, or 4 represent the size and extent of the primary local tumor. N refers to the presence or absence of malignancy in the regional lymph nodes. N0 means there's no nodal malignancy. With increasing N number, this would represent an increasing number of lymph nodes involved. M refers to the absence or presence of metastases. M0 means there is no presence of metastases. M1 and M2 may represent the type of metastases or location. You can see that this actually mirrors the mechanisms of cancer spread from the Cancer Spread podcast, such that the tumor or T stage is a reflection of the direct extent. The nodal or N stage is a reflection of the lymphatic spread, and the metastatic or M stage is a reflection of the hematogenous spread or metastases. Often, healthcare professionals combine this T and M staging into an overall Roman numeral stage, ranging from stage 1 to stage 4. Stage 1 tumors tend to be early tumors with no nodes or metastases. They are generally curable. Stage 4 tumors tend to be tumors presenting with metastases or advanced nodal stage, representing spread to other organs. They are usually incurable. Staging can be done both clinically and pathologically. Clinical staging uses the features of clinical exam or physical exam, as well as radiology and diagnostic imaging. Pathological staging assumes that the tumor has been removed, and often the local lymph nodes are sampled in such a setting, and this is how we get to pathological staging. Obviously, this is different than grade, as we spoke of before, which actually refers to the cells and their degree of differentiation and dysplasia, as seen by a pathologist, usually microscopically. This concludes our discussion on the important principles regarding the staging and grading of cancers. For further information, you can refer to the Basic Oncology Principles module and our video podcast about the different modalities of treatment on learnoncology.ca. Thank you.
Okay, that was a great little review on staging. I really liked their diagrams, but also very important for you to just know that information as you probably, if you even might be doing that in your clinicals right now, this will be helpful to you to know when you're looking at H&Ps of your patients. Next, we just want to go into genotyping. Your book definitely went into that. How it affects cancer diagnosis and treatment, because keep in mind, approximately 90 to 95% of cancers result from sporadic DNA mutations, including those that activate proto-oncogene genes to oncogenes. Some of these sporadic mutations are chromosomal translocations that turn on oncogenes. So cancer genetics also includes hereditary cancers in which a germline mutation in a tumor suppressor gene confers risk of cancer development in later life. Cancer-causing viruses can directly alter oncogene or tumor suppressive gene expression in the affected cell. Because of this, genotyping tumor cells at various stages of disease progression enables targeted therapies that can be very effective across a wide variety of cancer types. So in this slide, this is Dr. Turnbull. She talks about why genotyping is important in cancer care. It's very short, but again, great visuals, and she just does a beautiful job of explaining, how she uses it.
When we think about genomics in oncology, we need to remember that we're dealing with two different genomes. Firstly, there's the genome of the patient, and these are the changes that are largely present through nearly all the cells in the body. Changes in this genome we refer to as constitutional or germline variants or mutations. As clinical geneticists, our work is largely focused on using germline genomic variants to identify and best manage individuals at elevated risk of cancer. The second genome we need to think about is the genome of the tumour, and these are the changes that have arisen as that tumour has evolved, and we refer to these changes as somatic or acquired variants or mutations. These changes can inform on sensitivity to therapy and the prognosis of the cancer, and these types of changes have conventionally been the focus of the oncologist, the molecular oncologist, to define the management of that patient and cancer.
Isn't that interesting? It's a short video, but I just find the concept of genotyping it's so complex that just to learn about it in small spurts is probably the best way to try to understand it. I mean, there are tons of books on cancer genomics, so it's its own specialty. But the important thing is, as a practitioner, that the importance of it. And we talked about going through your patient's history and physical and doing that genome and drawing it out with the 3 generations, just to make sure you're not missing anything with these patients. Genetics are so indicative of like small changes in patients that can really help you guide your plan of care for them. So just be conscious of genotyping and how it's used in cancer diagnosis and treatment. Okay, next we're going into pediatric considerations. Your book talked a lot about leukemias, specifically acute lymphoblastic leukemia and acute myelogenous leukemia. They talked about bone tumors, nervous system tumors like neuroblastoma, retinoblastoma, and Wilms tumor. I just wanted you to be very conscientious about warning signs for childhood cancer. So obviously, if any of your pediatric patients have these things, or if you parents mention these things, this is when you need to seek medical early help for early ongoing symptoms. So white spots in the eye, new squint, sudden blindness or bulging eyeball, that's a definite. Any lumps or bumps any place in the body, but mostly on the stomach, the head, arms, legs, glands, and in the groin area, including in girls. Unexplained fevers, weight loss, fatigue, pale appearance, easy bruising and bleeding is a big one. Aching bones, joints, back, and easy fractures. Those are definite things to be concerned about, not only for cancer, but also if you're concerned about abuse. Those are things you have to be very conscious of paying attention to. Neurological signs, a change in walk, balance, speech, regression, continuous headaches with vomiting or enlarged head. These are all indications something's going on. So just keep in mind, children 0 to 14 years, that these are the percentages of cancers that are in the age group of 14 and below. The highest cancer is the acute lymphocytic leukemia, which is why your book goes into that. So make sure you're very well versed on that. And then in terms of adolescents, 15 to 19, the highest percentage cancer is the Hodgkin lymphoma. So just be very conscious of that too. Okay, so gerontological consideration. So as we know, during the process of aging, it also can affect not only our ability to develop cancer, but also our inability to respond to treatment if you did have cancer. So, aging is a funny thing because not only does it cause cancer, it limits the ability to treat cancer. And then to keep in mind also that cancer therapy accelerates aging. So it's a pretty wicked process when it comes to aging and cancer. So in this diagram, it really goes into like what happens at a young age and how your cells respond to cancer versus it happening at an old age. So it definitely has an effect. So the younger you are, in any diagnoses, it's easier to heal. Your cells have an easier job of healing. Unlike as you get old, everything slows down or decreases. Okay, so if you wanted to relook at any of these videos or look at any of these images, what articles they came from, all of the references are here, just so you know. I will be developing a study guide for our exam, our first exam that's coming up. It will be posted. I just have a few things to put together, but it'll be very helpful, and make sure you really review that in order to study for your exam. All right, guys, the next PowerPoint has more clinical information for your review, so please make sure you listen to that one as well. Thank you and have a great week.